Hi friends, so till date whatever slides I have presented, I hope that you have liked it. So here is another slide on biology. Cell organelles 2. First one I said about the cell organelles 1, right? In that I talked about the outer membranes, the cell wall and the cell membrane, details about that. This we will talk about the details of ribosome and mitochondria, okay? Mitochondria is hiding behind real mitochondria, okay? Cell organelles 2, ribosome and mitochondria. And I think these things you can see here. These are chromosomes, sister chromatids. The single part, let me tell you, we'll learn about DNA specially in another video because DNA is a large topic. We'll read about DNA differently, like DNA and chromosomes, proteins, etc. So for the timing, let me tell you, this total is considered as a chromosome, this X structure, and this single unit, this one thing, this is called chromatid. This is called chromatid, and this is called chromosome. This is chromosome, this is chromatid. Okay, and these two are called pair. When they pair, they are called sister chromatids. We'll learn about that. Just to represent this figure, I have told you. This is the nucleus. Inside the nucleus, it is there. You can see. This is the ER. This is the ACR. We have talked about partially about this in the previous slide while proving that cell wall is made up of peptidoglycan. So this is SER, this is RER. You can see ribosomes attached to it. Okay, these are mitochondria. We'll learn about that. These are Golgi bodies, most probably. Many more small things, lysosomes, etc. Many more vacuoles may be there. Most probably, I think it is a animal cell. Okay, so let us proceed. Now that day I escaped one topic from the beginning of the cell. I should explain it. It is the compound microscope, the microscope used for the measurement or the seeing the objects, observing the objects. One more microscope is there, that is the electron microscope. It was discovered in 1940 by Noll and Ruska. Okay, that is for more magnificently we can see. For the virus we cannot use this compound microscope. We have to use the electron microscope because they are more microscopic than the other organisms. Okay, so in this picture you can see the anatomy of a microscope. This part is the head, just representing it. This part is the head. It is the ocular lens. Ocular lens, just eyepiece you can say. Ocular means to notice, same as eyepiece. Diopter adjustment, the adjustment of the magnification of or the power of the eyepiece. For adjusting the power of the eyepiece, diopter adjustment. Okay, nose piece. These are all parts. The objective lens, this is also very important, through which the we see, actually we see through this, but the actual magnification happens here, right? This is the frame, a border type for the model. Coarse adjustment, fine adjustment, these are not so much required. While practical you can use, you must have seen this compound microscope in your lab. There's a light switch for on the light to be focused. Okay, and this is the mechanical stage on which the things are kept, the things are done. That's why it is called mechanical stage. Very important place to be done. Condenser we'll learn about here. Okay, condenser, illumination. Okay, and brightness adjustment. This is the base piece. Okay, this is nothing, this is the source of the picture. Then there's the stage control. How you can make this, the mechanical stage, right? It can be made down, it can be made up. That is controlled by this stage control. Okay, this part, right? Okay, not much required. Just this is very important. Magnification, how to calculate the total magnification. of a slide. How can you calculate the total magnification? Okay, how can you calculate? That is very interesting topic. PE into P O dot L. That means what? No one knows. I have written in short form. Power of eyepiece. Power of eyepiece. Okay, this I talked about the eyepiece. This part I talked about the eyepiece, right? That the eyepiece, the power of the eyepiece and the power of O dot L. O dot L means objective lens. Objective lens. So power of eyepiece into power of objective lens. This gives the total magnification. Okay. Suppose power of eyepiece is 30 times. Times is represented as x. You know that, I think. 30 times and this is 40 times. Total you can calculate easily. But while writing, see 30 times and 40 times. The times square it will not become. This is just to represent. Okay, we'll only just calculate 30 and 40. The total magnification will be, you can easily calculate that total magnification is equal to 1200 times. Times represent this, not that x square or times square. There is nothing. This is the just the how much times it is magnified. Okay, in terms of how much times, how much it is magnified, magnification is calculated. Okay, there's no much unit. Okay, now condenser helps to focus the light rays. You can see the condenser here just below the mechanical stage. This helps to concentrate the lights. Okay, helps to focus the light rays onto the slide and further to the objective lens so that it is more visibly applicable. Okay, you will see when you may have been shown by your teacher, biology teacher, that the many projections inside the through, maybe you have observed through the microscope. If you have observed, you have seen that some light is there. It is not totally dull. If it is totally dull, you cannot see that properly. You cannot visualize that properly. It has light, that's why you can visualize properly. Okay, let's skip this topic. Next. And now our next topic is ribosome. This is also an organelle of cell. Okay, let us see what is it. Ribosome. The first topic is only ribosome structure. Before that, I want to tell you something. For that, I am going. Wait. That for that I am going next. Okay. No, this I don't want. Oh, oh, yes. Some informations first to learn. Then you will go to the structure. What for that? You must know. Prokaryotic cell. You know the primitive cells. Prokaryotic cells have only cellular organelle, that is ribosome. They have only ribosome. The only cellular organelle that is ribosome. And this will study about what is 70s, what is 80s. Just remember, this is the fact, or this also is a differentiation point between the prokaryotics and eukaryotics. If question comes that difference between, tell the difference between prokaryotes and eukaryotes, you can give this point that prokaryotes have 70s ribosomes or ribosomal unit, and eukaryotes have 80s. But Swedberg will learn about that, ribosomal units, and they have no membrane. That is the most important thing. They have no membrane. Questions come like this, that name any membrane, no membraneless structure in the cell or organelle in the cell. So you can write this ribosome. One thing I want to sell not tell. Okay, cell structure and cell organelle, what is the difference? Cell organelle contributes to the development of the cell. something or the other, it lies within the cytoplasm. The main thing is that the cell organelles lie within the cytoplasm. Okay, they lie within the cytoplasm and become cytosol, right? Okay, so you know that cytosol is equal to cytoplasm minus cell organelles, right? Okay, so cell structure, the main cell structures are nucleus, plasma membrane, and in plants and fungi you can get the cell wall. Okay, so these are the structures. They are not directly related to cytoplasm, but organelles, ribosomes, lysosomes, mitochondria, we learned plastids, chloroplast, leucoplast, everything. Most part is in the cytoplasm only, right? So more number of organelles are there than the structures. Okay, and also they are the protein factories of the cell. They make proteins, they produce proteins. Okay, the main thing is only that their main function, the function of ribosome, it is, if it is asked, they help in the production of proteins. Okay, and it is found in almost all living cells, right? And this is a fact. These things you must know it. Discovered is very important. Short questions come from here. George Emil Pallade discovered it, and Richard B. Roberts gave this term ribosome. Okay, George Emil Pallade discovered it, and Richard B. Roberts gave this term. That's the ribosome. Okay, why given that, you ask him only? I don't know that. Okay, so we left some slides. Ribosome structure. This is the structure of ribosome. Okay, some part is hiding for my video, maybe. Whatever. Let us talk about the structure. This is the amino acid chain. Amino acid means the proteins, right? This is the amino acid chains linked with the what? Ribosome. Produced from the ribosome, right? A ribosome structure is only like this almost. Ribosome structure is only like this. Okay. Why? This is the large subunit. In my figure it has become smaller. Let us make it big. Okay, let us make it with another color. So somewhat difficult. Okay, so this is the large subunit, larger. This is the smaller subunit. There you saw. I will talk about it later on. For the timing, let us see. This is the larger subunit, the red one, and the blue one most probably is the smaller subunit. And between that is a chain of mRNA. Now, what is mRNA? This type of RNA. Right? It is messenger RNA. mRNA stands for messenger RNA. And also you can see the structure. This is the tRNA. That is the transfer RNA. mRNA, rRNA, tRNA. These are the main structures. mRNA means messenger RNA, ribonucleic acid. tRNA means transfer ribonucleic acid, and rRNA means ribosomal ribonucleic acid. It is mainly produced here only, rRNA. Okay, and this the growing peptide chain. What is peptide? Now you have to understand. It is also a chain of proteins, as it is said that amino acids is the same thing. Growing peptide chain. Peptide. I taught that day. Peptide, pepsinogen, peptosis. peptides, all these are proteins, types of proteins. So amino acid, from that only chains of amino acids form the peptide, and many peptides form the polypeptide, they form the proteins. Now let me give a brief explanation. Amino acid form the peptide, this form the polypeptide. These are not chains, these are just relation one by one, and this form the protein, right? Protein, right? Now, so many a chain of amino acids form peptide, a chain of peptide forms polypeptide, a chain of polypeptides form proteins. Okay, easy. So these are called the protein factories of the cell. Okay, so this is a chain of mRNA and this is tRNA. Nothing more you need to know. The groove between the two subunits furnishes the site for the synthesis of polypeptide. This is a groove. Groove, you understand? I hope you understand. Groove means where friction is created. That is physics part. Due to grooves and ridges, only friction is created, but that is not. Groove means any interaction part. Anything, any two surfaces, if they are meeting at a point, there must be some uneven surface. That is only groove. So this groove, this space provides the furnishes the site for the synthesis of polypeptide. This place, this groove provides the proper facility for the production of polypeptide, for the synthesis of polypeptide to form proteins. That's the thing. Now I was talking about the unit. S is the unit. It stands for Swedberg. Again I am drawing. This time I will draw bigger. Okay. So this is the larger subunit. This is the smaller subunit. Okay. So suppose this has a power of 50s. 50s. 50s, right? I think you can understand. This is a power of power or not. It is of the unit 30s. Okay. So this will comprise of what? This will comprise of 80s. So where we saw 80s, we saw in the eukaryotes, 70s in the prokaryotes and 80s in the eukaryotes. They have more ribosomes because they are more functional. Eukaryotes are more functional, that's why they require more ribosomes. So these are the divisions. Like this they are divided. In the larger subunit some part is there, and the smaller subunit some part is there. If you add them up, you will get the total part, and that is how the ribosome units are calculated. And this S stands for Swedberg, is a name of the person who invented it, Swedberg. Okay, many students write the SVEDBURG, but it's not BURG, it is BERG. Okay, just remember it. I also sometimes make mistakes, that's why I'm saying. Okay, unit, it is a unit to measure in terms of, in terms, okay, you need to measure in terms to measure, you need to measure in terms means in terms of something it is measured. Every measurement is there for something. Ribosome, there is no length, mass, what you will calculate. So in terms to measure sedimentation rate, the sedimentation of proteins take place here. That's why they are called the protein factories. While the synthesis of proteins, the sedimentation is going on. So the sedimentation, how fast it is, the sedimentation rate of each unit is calculated and then added up for the whole unit. Okay, Whole unit it is, for I said that both you will add up and get the total. But for the sedimentation rate of proteins, it is used. Okay. Why are most ribosomes attached to RER? This is a very clear fact. ACR is smooth and RER is rough because of the presence of ribosomes. Most ribosomes are present on the RER. Some are free in floating in cytoplasm, but most are here. Okay, so proteins there. So what benefit they get? Why are they attached so much? Because the proteins directly go into RER, means proteins are manufactured from the ribosomes, and if they directly go into the RER or ER, whatever you say, endoplasmic reticulum, rough endoplasmic reticulum, that I should say, then they will be safe from other enzymes of cytoplasm. Cytoplasm contains many harmful enzymes, maybe. So they should not go into the cytoplasm. It may harm them. So for the protection of the proteins, rather I should say, they are going directly into the ER, and from there they are transported through the ER. So that will be more beneficial. This way you have seen, right? Ribosomes. Okay. So next topic is mitochondria. Mitochondria is generally the energy powerhouse. For generating energy, it is used for generating energy. Okay. So mitochondria, some facts are there to figure out. It was observed first in insect muscle cell. Insects have also muscle. Okay. So in insect muscle cell by Colliker in 1850. Okay. 1850 by Colliker. Often concentrated in more active regions. Why? I said, see, they are called the powerhouse of the cell. They are mainly used for production of energy. They are helpful in only production of energy. Their main function is to produce energy. So they are more present in active regions like muscle cells. Muscle cells, you have to do more muscular work. So energy is more required, more force. If you have to apply more force, then you have to gain more energy, cellular energy. So that is provided by mitochondria. So it is more present in active regions. They are called the powerhouse of the cell. They produce energy in the form of ATP. The energy is produced in the form of ATP. ATP means adenosine triphosphate. It is generally produced from ADP, adenosine diphosphate. Okay, one more phosphate ion is added, and that becomes adenosine triphosphate. Okay, so the energy in this mitochondria is produced in the form of ATP, and as this is the main unit, means of energy in the cell. This is the unit of energy in the cell, ATP. So that is also called energy currency of the cell. Like you have 10 rupee note. You have 10 rupee note. So that is the currency. So from that you are buying something. So that will be helpful. But the thing from which you are buying, that is the currency only. So here also it is the energy currency of the cell. In the form of ATP, energy is produced. So ATP is more important here. Okay. Also, if I think you have heard this, I do not have to tell again and again this, that plastids and this mitochondria are called semi-autonomous organelles. Now you may ask that who are autonomous, then fully autonomous? It is nucleus. Nucleus is called the brain of the cell, so it is fully autonomous. Okay, it is not controlled by anyone. It is controlled by itself only. Okay, so it is mitochondria is called semi-autonomous because it has its own DNA and ribosome. Why it has its own DNA and ribosome? Why so partiality? Why Golgi bodies don't have? Why plasma membrane don't have? Why lysosome don't have? Why they have? Mitochondria means ribosome is very small organelle that may be consisted in many things because they are the protein factories. All cell organelles may require protein factories. So mitochondria is more bigger unit than ribosome. Okay, so semi-autonomous. Semi. First, let us break the semi. Why? Because, let us break the word autonomous first. as it has its own DNA and ribosome. Some power is it with him. Let us first not get up to this. Let us see from here again. Why semi? Also semi because still some enzymes. It is enzymes. Okay, it's not there. Still some enzymes are controlled by nucleus and other organelles. Still some enzymes are controlled by nucleus and other organelles. There are some other organelles also. The nucleus is structure, and that's why other organelles is said. We can say that controlled by organelles like nucleus. That also we can say, but here it's not said because nucleus is structure. I explained what is structure and organelle. What is the difference between that? So it is semi because some enzymes which it is controlled by lies in the nucleus or the other cell organelles. Okay. So why, but autonomous because it has its own DNA and ribosomes. Why it has? Because while the development of cell from the prokaryotic cell to the eukaryotic cell, the DNA and ribosomes were inorganizingly spreaded, or you can say they were unorganized. Here and there they spreaded. Some remained DNAs and ribosomes, some remained in the cytoplasm, some remained in some other organelle, some went to the nucleus. More part went to the nucleus. So this mitochondria and plastids during the development of the cell, they had a great part of this DNA and ribosome due to the unorganizable spreadment. Okay. Also, it is double membrane. There is an inner membrane and the outer membrane. So it is double membrane structure. Okay. Now, this was the informations. This is the structure of mitochondria. You can see here. These things were not there. That's why I have given here. Inner membrane. This is the inner membrane. This structure you can see. These things, and cristae are the folds. Cristae are the folds. You can see these folds. These folds are called cristae. Okay. This part, inner part between the folds, is called cristae. These folds are called cristae, and these are present in the inner membrane. Okay, inner membrane you can see these structures. Okay, not within the outer membrane. This is the outer membrane. You can see this brown color structure, and this is the inner membrane, right? This is the inner membrane. This is the outer membrane, and the space between the inner membrane and the outer membrane is called perimitochondrial space. You can see here perimitochondrial space. And the matrix is what? It is a fluid-like substance which is present inside the mitochondria, means inner membrane within the cristae. Okay, this blue color structure which is spread is fluid-like substance, holds the oxysomes, F0F1 complex within it. What are F0F1 complex? These structures, this pin-like structures coming out of the cristae, they are present on the cristae only. This F0F1 complex, also called oxysome. Okay, we'll learn about that. They are present on the base only, of base of cristae. In the inner membrane they are present. They are, they actually have main help in the ATP production, is the energy production. This part is only the helping most. Okay, they are only the structural units which help more. Because of them only the energy is produced. This is just the simple structure of mitochondria. This is the structure of oxysome or F0F1 complex. You can see here oxysome or F0F1 complex. Why it is called F0F1, I will tell you. Oxysome. Okay. In the mitochondria, the energy is produced with the help of some enzymes. Some enzymes. Yes, this thing is there, but some more enzymes are present within the matrix. That is the one of them is of the Krebs cycle. We'll see it of the Krebs cycle. That is the oxidase. Oxidase is a enzyme oxidase. Okay, oxidase is a enzyme which is due to this special enzyme, the energy is produced in the form of ATP with the help of some special enzymes such as oxidase, you can say, while writing the answers. So this is one of the enzymes present in the matrix. Okay, so because of this enzyme only, which is helping in the ATP production, especially these structures which are main parts of ATP production, they are called oxysomes. Okay, because of this oxidase, as it is a very important enzyme of the matrix, and in the matrix, in the cristae, on the base of the cristae in the matrix, this F0 F1 complex are lying. That's why it is called oxysome. So it is a headpiece. This circular part is a headpiece, and the stalk-like structure is called stalk or pedicel. Any one name you can give. This is a base piece. So the line is not given, the points are given. Whatever. This is the base piece. Okay. So it generally looks like a F0 F1 complex. How? If I take you back to the slide, this slide, in these structures, you can see which is to the base of the cristae, this stick-like structure. That means, and here also is a base is there, right, at the bottom. It's not visible with the microscope, but electron microscope you can easily visualize. So this is the structure, right? Here the cristae is there, right? And here matrix is there all around, fluid-like substance. So the base is connected with the base of the cristae, right? The base is connected with the base of cristae. The base of cristae. Okay. Base means, okay, let me go to the next slide. Explaining here. So this is the this is the structure. Base is there. Here cristae base is there, right? You have learned, and not learned, you have heard. G plus 4 flats are there. G plus 4 flats are there. That means ground floor. G for ground floor, sometimes garage. Okay, ground floor specially, I should say. Generally counting any building. Suppose this is a building. This is 1 story, 2 story, 3 story, 4 story. This what we'll say? We'll say first floor. No, this will say ground floor. And this we say first floor. This we say second floor. And this we say third floor. Right? This is very important concept why it is F0 F1 complex. Right? So we start with G. Okay, and start, then next one is 1. Okay, so the base of the cristae is equal to the base of the oxysome. Okay, so base means G, ground floor is also called the base floor. I think you know that's very easy topic. Ground floor is also called the base floor. So the ground floor matches with the base floor of the cristae. So that means this base piece and this top piece, as this look like the same, like a rectangle, this can be considered the base piece, and this can be considered the first floor, this can be considered the ground floor, this total, and this can be considered the first floor. And before 1, what is there? Base floor. So before 1, what is there? What comes? 0 comes. 0 comes. So can't we call this F0 unit as it is the base, it is on the base of the cristae, and we start generally with the ground floor or the base floor or the zero floor, and next comes the first floor. So can't we tell this structure as the F zero floor? Can't we? We can. F is generally a unit who in, first saw it, Paliker maybe. He named this F. There is nothing, just to make you understand the digit. Why this is F0? Why this is F1? People can think this looks that people can think that this looks like a zero. That means it is F0, right? F0, and this looks like a one. See, total one digit only we can see here. So it is F1. That is the most common mistake, but this is not correct. This is not correct. Why? Because it is the base piece, and generally the base, and it is matching with the base of the cristae, and generally the base we start from ground floor or base floor or zero floor. So F0, this is unit, and this is the F1 unit, the first floor, right? It is totally different in shape, and these two are same in shape. That's the difference. Headpiece, stock or pedicel, and base piece. This is the F0 unit and this is the F1 unit. This total structure is called oxysome or F0 F1 complex. You will get the term most probably F0 F1 complex in most of the books. That's why I've explained why F0, which is F0, which is F1. Okay, we're almost to the end. More information on the mitochondria. Yeah, saw the cristae. These are the folds, right? Cristae are the folds. A straight line and the folds. These. Which distance will be more? Of course, the distance in the curved part will be more because it is bending in different directions. It is bending, right? So the curved path, the more distance will be there than the straight path. So as more cristae are there in the inner membrane area, so more the presence of more cristae, this means increasing. This sign means increase, right? So as more cristae is there, so inner membrane area will increase. Cristae are present in inner membrane, so its area will increase, and there only in the to the base of the cristae oxysome is there. So more the cristae, more the area, more will be the oxysome, the space for the oxysome, and more than we know that for oxysome only ATP production is possible. So more the number of oxysomes, more ATP, and thus more energy. Okay, so cristae is very important part in mitochondria, and oxysomes contain ATP synthesis. This is also an enzyme. Because of this enzyme, mainly the ATP production is possible. Oxidase, I said in that was in the matrix. Okay, all enzymes are required, but this is most important. Okay, now cristae structures are also different. They vary in number also. Okay, so the structure in animals most common is lamellar, and in plants most common is tubular. Lamellar, you can say like this, or sometimes round, sometimes disc, some cross. Anything happens that structure. But tubular, you know that the microtubules also, tube like cylindrical tubes. It is like in the cylinder shape. Okay, okay. And matrix contains enzymes of Krebs cycle. It is also called the citric acid cycle also, you can say. It's a very important cycle in the production of energy for the mitochondria. So it contains various enzymes of Krebs cycle. Is one of them, you can say that oxidase, and also many DNA, RNA, proteins, all of the like in the cytoplasm everything lies organelles. So different parts of mitochondria also lie in the matrix. That is the fluid substance. Like in nucleus, nucleoplasm, the DNA lies. Here also in matrix, DNA lies. Proteins, lipids, etc. Okay, so two types of proteins here is said. These are two types of proteins here. As you can see here, I have mentioned. These are two proteins. Porins and permisses. Porins are the proteins which help to make channels for free movements, while the opening of the plasma membrane, when the plasma membrane opens for the free movements for any cellular organelle or proteins free movement for themselves. This helps to make channels for free movement. Okay, and the permisses are the proteins which help in active transport. We'll see the active transport slide also. It is the movement of ions or molecules, and it requires energy. Okay, active transport, so it helps in active transport. Sodium ion, potassium ion through in and out of the cell. Okay, that is only the thing. Now most probably you'll get the Krebs cycle. I'm not going into detail, just to make you sure what is it. Okay, we can see here the pyruvate carboxylase. You can study it more detailed in the internet. You will get many things. If you are of higher classes, you can easily understand it. So it is nothing. You can say it is citric acid cycle also. Krebs cycle. See, NADH, it is a very important thing which is formed here. Also, sometimes ADP, then PI, you know, inorganic phosphate, right? PI means inorganic phosphate. So sometimes ATP. These are very important terms you must know at this time. This is a Krebs cycle, very easy cycle. You can go in detail. I am not going in such details of these terms: isocitrate, dehydrogenase, and ketocurylate, etc. Hydrogenase. All these are hydrogenase, hydromaterials, dehydro substance, no water. But here some water is coming. Cycle means only repeating the same thing. Some stage it went, goes, and then it repeats. Acetyl is formed here. Acetyl N-acetyl glucosamine. Not, it is not that. The main thing is here only the citrate synthesis. Okay, we talked about the ATP synthesis. That is the citrate synthesis. So in the Krebs cycle, it is citrate synthesis. But in our mitochondria, the main cycle, that was the ATP synthesis was formed. Okay, from that energy ATP was formed, and from that energy was produced. Okay, just simple. It is very simple. You can study more. You will see this slide also again. Now we are almost to the end. So this is active transport. You can see the sodium Na plus, sodium ion, potassium ion, concentrations here, the levels is given. See ADP, I said adenosine diphosphate, and this ATP, adenosine triphosphate. See ADP is combining with phosphate, one of the phosphate. Adenosine, pentagon structure, pentose sugar you can say, and these are the two phosphate ions. Previous slide also I said what is phosphate. Symbol PO4 minus, PO4 negative. So these are two phosphate ions, that means adenosine diphosphate. Adenosine, this is adenosine, this is two phosphates, diphosphate. Di means two, and it is combining with one more to form ATP. Adenosine this is, and this is three. Tri means three phosphates. This helps in the passing of the molecules or ions through the, this is the plasma membrane. You can see the hydrophilic and the hydrophobic region. This is the hydrophobic, this is the hydrophilic. All is taught in the previous video. You can see this, it is opening, and this potassium ions are coming, and also the out also it is going. This is the extracellular space, and this is this inside it is cytoplasm. From inside to outside it is also going, and from outside to inside it is also coming. So here you can see the definition of active transport: the movement of ions or molecules across a cell membrane. A cell membrane is required here. So the movement of ions or molecules across a cell membrane into a region of higher concentration. And the main difference between the two types of let me just explain it. The two types of transport are only passive transport and active transport. Let me differentiate. Passive transport, a region from higher concentration to lower concentration it goes. Active transport, from lower concentration to higher concentration. This is only the main difference. And one more difference: passive transport do not require any energy, but this active transport requires energy. So it is from low concentration low concentration to high concentration, right? Low concentration to high concentration, and it requires energy. This is the main differentiation point between passive transport and active transport. Passive transport and active transport. The movement of ions or molecules across a cell membrane into a region of higher concentration, assisted by enzymes. Enzymes help in it, and requiring energy. So when it is going from here to there, that means from a region of lower here, then is density. Density. I'm saying deficiency. So here is excess. It is going again, and when it is feeling that here nothing is there, then again it coming. Okay, so lower to higher region. That's why many passages are there of lipid bilayers. So these are all this thing is only lipid bilayer. So from that it is always continuously passing. So there is no thing that it is empty. This place is totally deficient. Never. If from here it is going to lower to higher concentration, then again it is coming from lower to higher concentration. The sodium ion may be lower, this may be higher, potassium ion. So it differs. It is not static thing. Okay, I think I took much of your time. So this is the difference. Last slide is inner membrane and outer membrane. Okay, inner membrane and outer membrane. So what is there? Nothing. The inner membrane of the mitochondria and the outer membrane of the mitochondria. What is the difference? 20% lipids, and it is composed of 50% lipids. So what is the 80%? 80% proteins and 50% is proteins. So what are the proteins? Permeases in case of inner membrane. It helps in the active transports. That means inside the inner membrane also active transport take place, as because outer membrane is not so much rigid. Okay, you can see here that it is freely permeable and is semi permeable. It allows selected materials to come through, pass through. That means what happens, later on I'm telling you. So this 50% proteins, and these proteins are porins. Outer membrane proteins are porins. Okay, this is rich in enzymes. Many enzymes are there within the matrix, and this lack of enzymes, there's nothing is there. Okay, in contact with cytoplasm, with the outer, you can see this is the outer membrane, this is the inner membrane. This is common thing. There is the matrix, right? So this membrane is contact with the matrix. That means the inner membrane, and the outer membrane is contact with the cytoplasm. Outside cytoplasm is there. This is the mitochondria, right? This is the mitochondria. So outside is cytoplasm. Mitochondria is there in the cell, in the cytoplasm. So in contact with matrix, in contact with cytoplasm. And here more surface area due to the cristae, and here is less surface area, and it is the outer membrane is somewhat smooth, and inner membrane is rough because of high number of proteins. I said in the RER also, because of the presence of ribosomes which produce proteins, they are rough. So it is rough because of high number of proteins. Why high number of proteins are there? Because cellular respiration takes place here. Because of this cellular respiration only, the energy is produced in the form of ATP. Now I was telling that if it is freely permeable through the outer membrane, this is the outer membrane and this is the inner membrane, right? So outer membrane, it is freely permeable, anything can come here, but it allows the inner membrane is semi-permeable or selectively permeable. It allows very or differentially permeable. It allows very selected materials to pass through it. So what will happen to the rejected materials if inner membrane selects some material? Suppose A and B, A plus B come through outer membrane. The outer membrane is freely permeable. Anything you come inside. He is open-minded, open heart. So he allows everything. But inner membrane is saying I will only take A and I will not take B. I will not take B. So what will happen? What will B do? It has entered through the outer membrane, but it is inner membrane is not giving the passport that yes you can enter our country. Is not applying the visa. So what you will do? During the studying the structure of mitochondria, we find that here is the mitochondrial space between the two membranes, between the outer membrane and inner membrane. This rejected materials by the inner membrane stays in this perimitochondrial space. Okay, most probably that's it. So thank you and hope I would you would like it. Please subscribe my channel and like this video. And for any recommendations or suggestions or better something, you can write to me a comment in the video. I would readily accept it. Okay. So thank you for watching my video. That's it. And hope I would come with more better videos and shorter time. Today was a bit longer. So bye. Bye. Uh, auf Wiedersehen. Let us say auf Wiedersehen is goodbye in German. So auf Wiedersehen. Bye.